group to lead us into prayer.
చాలునయా చాలునయా నీ కృప నాకు చాలునయా నీ కృప నాకు చాలునయా 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 నీ కృప నాకు చాలునయా నీ కృప నాకు చాలునయా ప్రేమామయుడివై ప్రేమించావు కరుణామయుడివై కరుణించావు ప్రేమామయుని ప్రేమించావు కరుణామయుడివై కరుణించావు తల్లిగా లాలించి తండ్రిగా ప్రేమించి తల్లిగా లాలించి తండ్రిగా ప్రేమించి ప్రేమ కరుణ నీ కృప చాలు ప్రేమ కరుణ నీ కృప చాలు చాలునయా చాలునయా నీ కృప నాకు చాలునయా నీ కృప నాకు చాలునయా.
Let us do our daily prayer. Our Father who art in heaven, hallowed be thy name. Thy kingdom come, thy will be done, on earth as it is in heaven. Give us this day our daily bread. Forgive us our trespasses, as we forgive those who trespass against us. And lead us not into temptation, but deliver us from evil. Amen. O most merciful God, grant me, I beseech thee, ardently to desire, prudently to study, rightly to understand, and perfectly to fulfill that which is pleasing to thee for the praise and glory of thy name. Amen.
Thank you, dear students, for the meaningful prayer and invoking God's blessings. The welcome speech is that allures the attendees to either listen and take interest. The opening speech sets the tone for the entire event. Today, we have different age groups attending the seminar. With a warm heart, we want to welcome all of them. I invite Dr. K. Swarnalata, Associate Professor, Department of Chemistry, to welcome the gathering.
A very good morning to one and all. Coming together is a beginning. Keeping together is a progress. Working together is success. Fresh beginning always exist. We have to welcome them. To find God, you must welcome everything. It's my immense pleasure to welcome our dear superior and correspondent, Dr. Reverend Sister Marita Pudota, Sister. Welcome, Sister. I am very much delighted to welcome our dynamic principal, Dr. Reverend Sister Marita Di Mello. Hearty welcome, Sister. I would like to extend a special welcome to our today's resource person, Dr. D. Madhavi Garu, founder, Institute of Biochemical Technology, Hyderabad. We are extremely happy to have you, Madam. Hearty welcome. I heartily welcome Dr. Reverend Sister Shaurilu, Vice Principal PG Directress, to this webinar. Hearty welcome, Sister. I want to extend my heartfelt welcome to all the HODs of the various departments. Welcome, madams. It's my privilege to welcome all teaching staff and supporting staff. I am very happy to welcome our dear students for this knowledge-oriented webinar. Welcome one and all.
Today we have in our midst an young researcher and scientist, one of our proud alumni, who completed intermediate in the year 1992 and graduation in 1995. He is none other than our resource person today, Dr. D. Madhavi. An introduction can help establish credibility, especially, but also trust and likability. The result of a good introduction is to greatly increase the likelihood that the speaker will do well and feel comfortable with the audience. Now, I take this opportunity to welcome Dr. Gayatri Devi, Associate Professor, Department of Chemistry, to introduce.
It's my privilege to introduce Madhavi ma'am to all of you. As Henry says, patience, persistence, and perspiration make an unbeatable combination for success. These words are true to our esteemed guest, Dr. Madhavi Doma. She has completed her post-graduation in MSc Chemistry, MSc Plant Science, and Bachelor of Education. She received her doctoral degree in genetics. She qualified CSIR fellowship, JRF, SRF, and national eligibility test. Dr. Madhavi ma'am is having 18 years of research experience in both genetic engineering and chemical engineering. She has 8 publications and 1 patent filed. Her interest in research made her to travel far and wide to become the founder of Institute of Biochemical Technology. She drafted a new subject, Natural Product Technology, for MTech Biotechnology from an university in AP. She is also Boards of Studies member to various universities. Her interest in community services, she got NSS certificate for adult education service. Welcome, madam.
Thank you, madam. Yes, madam. I want to put before you a few things. I'm
Excuse
very
me.
happy. Am I audible, madam?
One more minute. I want
Ah,
to give a brief introduction about the topic. It makes you easy. Before going
ah.
into the talk, I take this opportunity to thank Gayatri ma'am for introducing a chief guest or the resource person. I take the opportunity to give a brief introduction of the topic for today. Natural products technology. The importance of natural products for medicine and health has been enormous. The novel COVID-19 pandemic, starting from December 2019, has caused unprecedented threat to public health worldwide. We called attention to natural products and herbal medicines for prevention. We started using home remedies like ginger, pepper, tulsi, mint leaves, etc. So, what are natural products? A natural product is a chemical compound or substance produced by a living organism that is found in nature. Natural products can also be prepared by chemical synthesis and have played a central role in the development of the field of organic chemistry by providing challenging synthetic targets. The term natural product has also been extended for commercial purposes to refer to cosmetics, dietary supplements, and foods produced from natural sources without added artificial ingredients. Within the field of organic chemistry, the definition of natural products is usually restricted to organic compounds that are isolated from natural sources that are produced by the pathways of primary and secondary metabolism. Although natural products have inspired numerous drugs, drug development from natural sources has received declining attention in the 21st century by pharmaceutical companies, partly due to unreliable access and supply. Drug discovery from natural products requires a multidisciplinary approach, utilizing available and innovative technologies to package such natural product compounds for medical practice and drug development. Natural products derived from plants and animals play important roles in shaping modern medicine. New chromatography and metabolomics method, together with the expansion of natural product databases, are enabling sensitive and rapid identification of compounds with medicinal value. Today, we are really fortunate uh, to have Dr. D. Madhavi, who is the young researcher and the scientist who is doing enormous research work in the field of medicinal plants. Now, I hand over the session to Dr. D. Madhavi, our resource person. Welcome, ma'am.
Thank you, ma'am, for the introduction and for the prayer also for the students. I thank. And also, I'm very much happy. I'm very much happy to give an online lecture to the students where I have studied, and I'm very much happy that my students, my college is coming before me and giving some introduction about me and everything. I'm very much thankful to principal sister also, and for giving me this opportunity to keep before you few things about natural products and the techniques involved in it. So, and I hope that everyone is fine in this pandemic situation. And I want to continue the natural product technology online session. Shall I start, madam? Madam,
You can
is
start,
it
ma'am.
visible, madam?
Yes,
PPT
yes,
visible,
ma'am.
madam? Okay,
Yes,
madam.
yes,
Thank
ma'am.
you very much. Uh, I'm continuing, madam. Yes, and
Sure, sure.
this
You
is
can continue.
okay, madam. Thank you, madam. And here I'm Dr. Madhavi giving online lecture on natural product technology. And the concept is under the natural product technology, the concept, a small concept is isolation of pure molecules from natural sources and techniques involved in it. And I'm here. I'm the founder of the Institute of Biochemical Technology. On behalf of the Institute, I'm delivering this lecture. And yes, everybody knows that at present, every scientist throughout the world, they are recognizing the value of natural products, how they are interfering in our metabolisms, and how they are curing the diseases, how they are satisfying the human needs. And previously, 1 year back, actually, the natural product is a very big technology that is not identified by many scientists, and it is due to the cost-effective nature. Many left over this complete subject. So I want to revive it. I want to encourage the students to take part in this natural product drug discoveries, so that I will introduce few techniques how to isolate the molecules and how to identify the molecules and what are the techniques involved in it. I want to put before you all this new information, whatever I have identified till now. Yes, madam. IBCT is the first private institute in India to offer training in genetic engineering technique called hairy root induction. Yes, it is a broad concept. Natural product technology is the combination of different techniques that belongs to multidisciplinary sciences. That is genetic engineering, microbiology, biotechnology. Every technique is involved related to the natural products. That means not only till now we have an opinion that natural products is nothing but it belongs to organic chemistry. No, in through genetic engineering also we can enhance the molecules through with the help of biotechnology and everything. Because as the natural products are available in the plant system or animal system or some or a microbial system, very less quantity. So, in order to enhance these molecules, genetic engineering tools are required, biotechnology tools are required. So, that concept is also included in the natural product technology, and IBCT offers all this training, online training and also offline training in the hairy root induction technology. This technology is slowly gaining importance. And IBCT is the first private institute in India to isolate anti-cancer gloriosin and anti-cancer 3-D-methyl gloriosin from Gloriosa superba lineus tubers. Yes, why I am stressing these two molecules is since in 1952, these two molecules, anti-cancer molecules, are identified in 1952. But till now, Nobody isolated these molecules in India. So IBCT for the first time isolated these two anti-cancer molecules, and I encourage researchers and scientists or the scholars, research scholars, to take up these molecules and to trigger the research on these two molecules because of because of the properties that are identified previously. Till now, preclinical trials and clinical trials of these two molecules are not yet over. So, due to unavailability of the molecule, so now you can start doing some research on these two molecules, and IBCT will help in guiding their work on gloriosine and 3D methyl gloriosine. And it is the first institute in India to offer theoretical training in media manipulation. Yes, media manipulations are very, very important to enhance the quantity of the natural product. And here in this picture, you can see how the media is helpful in the culture of the different transformed cultures. Here the growth of the culture is very less, but here in second and third bottle culture, the growth of the culture is more. This type of media manipulations is standardized by the IBCT. This type of technology is offered in IBCT for the first time. And I want to go into the subject that what is natural product and what is isolation of the compounds. Yes, the natural product technology is a vast, a wide area which occupies wide area in organic chemistry and also genetic engineering. And under this concept, isolation of the compounds comes. The subject which deals with the immense potential and isolation, enhancement, bioactivity testing of natural products isolated from microbes, plants, and animals. We have an opinion that plants might. We have an opinion that that natural products are isolated from plants. No, these products can also be isolated from microbes, animals, and other marine organisms. So whatever the molecule that is isolated from a natural source is nothing but a natural product, and the technology or the techniques involved to isolate a single molecule or to test a single molecule is nothing but the natural product technology, the complete entire information. The current online lecture is on the topic on the prime concept isolation of commercially useful natural products from plant sources and techniques involved in it. Yes, I'm stressing especially the small concept that is commercially useful natural products, that is in the view of pharmaceutically useful natural products. Natural products are useful in many ways, that is as a food material, as a nutritional material, like that. But here in this concept, how to isolate pharmaceutically useful bioactive product from the natural system. For I have taken an example from the plant system. In this lecture, examples are given from the plant systems. So marine organism systems are there, microbial systems are there, animal systems are there, but I'm stressing especially because this is many more examples and it is easy to understand also. Some of the natural products are well known to every student. So I'm taking the example of plant natural systems. So the molecules isolated from plant systems which are useful pharmaceutically. That is the main concept. Natural products play a key role in many fields. One of the important field is pharmaceutical field, as I have already said. Therefore, for the isolation of the pharmaceutically important molecule is dealt in this concept. And the magnificent role played by the natural products. Why only natural products? Why not synthetic products? Generally, we all know since ages, our human beings are using for all health needs natural products. And what is the main role played by it? Why we are not choosing? Why the synthetic products we are not choosing to use as a pharmaceutical molecule? So now, what is the importance of that? Using natural products shows less side effects and very effective in curing diseases. That is the main concept why we are stressing the need for natural products now. In this pandemic situation, it is highly needed. The research of natural products is highly needed to identify new effective drugs. Already there are drugs identified in the natural systems, very few drugs. There is a need because this disease is a new pandemic and there is a need to develop new molecules from the new natural sources. And it is, I want to give a good information that only 5% of the plants are explored till now. 95% of the plants on earth are not explored. Here we are facing the pandemic situation. That means a new disease. We are encountering a new disease. That means it is also needed. New molecules are needed to encounter, to face, to cure the disease. So that means new discoveries are needed. New plants are should and must be explored so that we can identify new molecules so that to encounter this new disease. Not only now, in future also, if there is any pandemic like that, definitely there is a solution. There is a ray of hope that we can find the solution in the plants or in the bacteria or in other organisms. Definitely it is sure. Because why? Because the reason is natural products are structurally optimized by the evolution to serve the particular biological functions, including the regulation of endogenous defense mechanisms and interaction with other organisms. What exactly it is the products evolved in the plants, or the products, the antibiotics or bioactive molecules evolved in the evolution in plant system, go hand in hand with the evolution of other organisms. Yes, I want to put in easy terms that if there plants develop molecules to attract insects, that means we will come to know that insects are already evolved at that time. So in order to attract those insects, it developed different molecules. That means the development of the molecule is going hand in hand with the evolution of other organisms also. So these molecules are optimized. That's why these molecules also shows less side effects than the synthetic molecules. And historically, natural products have played a key role in drug discovery. Yes, at present situation, in previous also, about the molecules we are using therapeutically or pharmaceutically or either natural products or the models of the natural products. So since ages, we are depending for everything on plants or other organisms. Pharmaceutically also, we are depending on plants and other organisms. So historically, they have played, already we have visualized all these things. It played a key role in drug discovery also. To discover a drug synthetically also, these molecules will play a very good role. By taking the model of a natural molecule, we will design the other molecules. Oh, this molecule is showing less side effects, so we can synthesize this molecule. It is also an initiation step. Discovering this molecule is an initiation step to discover other molecules synthetically also. And another important point here is the natural product pool is enriched with bioactive compounds covering a wider area of chemical space compared with the typical synthetic small molecule libraries. Yes, it is very clear that this occupies a wide area. That means many molecules are discovered with very complex structures and also simple structures that are active on many diseases. A single molecule shows a very good activity on not only one disease but many diseases. And moreover, synthetic analogs of natural products with improved potency and safety can be prepared with, and therefore natural products are often used as starting points for drug discovery. Yes, the drug discovery is a very big concept, and there are many steps involved in it. Many techniques and many technologies are involved in it. And this is the natural products are playing a key role for the starting points for drug discovery. And numerous drug discoveries it inspired. So the above facts are strongly encouraging the world scientific community to explore the new molecules from natural sources to extend our healthy life. Why we discover all these molecules is just to extend our healthy life, our healthy life. The above facts. And what is the current scenario? Why we have to learn all these techniques? What is the current scenario of the natural products in the world? According to WHO, World Health Organization, at present, even today, 70% of the world population is depending on herbal medicine for their health needs. Yes, it is true. And WHO reported in 2018 that 88% of all member states acknowledged the use of traditional and complementary medicine. This is the report given by WHO. And even in our country also, Ayurvedic medicine is very popular, and 70 more than 70% of the population in India are depending on herbal medicines and other medicines that are acquired from that are obtained from the microbes and also other organisms. That means About 70%, 70 plus percent of population in India are still dependent on natural products. A large number of currently prescribed drugs have been either directly derived from or inspired by the natural products. And there is an important thing, this information, what is the natural product technology information, is not it is not a one day research or 2 days. Since ages we have developed a that means methods and identifications and everything, and all this information our previous generations compiled all this information in the text, and they have given to the future generations. And what is the method they have followed is trial and error method. At that time there are no scientific methods, so they use a trial and error method, and with their experiences, with their observations, they have compiled all this information. And one of the very good information is the Ayurvedic text. In these texts, very good information is given about the plant extracts and which plant acts on which disease. This information, we can grab the information and we can do further isolation of the molecules. So, in these texts, since generations, trial and error method, what they have used is also a very good method. And this method we can believe and we can go further by using this information on plants. Extracts of some natural products led to the modern discovery of their active constituents and eventually to the development of new drugs. Yes, we have come to the concept in the natural product technology, pharmaceutical sciences, and the development of the new drugs. Under the development of the new drugs, isolation of the pure molecules from the natural sources will come. And what are the steps? Simple, what are the steps that are there in the development of the new drugs? How we develop a new drug? First step is isolation of pure molecules from natural sources. Next one is bioassays. Next is toxicology studies. Next is preclinical, and fifth is clinical trials. We cannot say that a drug is developed till we complete all these steps. The first step, I'm stressing the first step because remaining steps, there are experts and everyone who knows better in these subjects. Isolation of pure molecules from natural sources is the first step in the development of new drugs. And isolation of the molecules. In the isolation of molecules also, there are few steps involved in it. The first step is literature survey and also knowing the complex structure of the molecules and its uses. This is the first step in the isolation of the molecules. Without literature survey, without previous texts, without going through the scientific reports or publications, we cannot do anything further. We cannot isolate a single molecule. So we must and should thoroughly go through the literature survey. That means what we know through the literature survey, how it is useful to isolate a molecule. This concept, these points are stressed here, and in this slide it is clearly given what we know in the literature survey. Before going to isolation of the molecule, these all points we have to learn. What we know in the literature survey, we are going to know availability of the material. Yes, when we study the scientific reports or the reports of or the Ayurvedic text or some other information through other persons like that, we will come to know whether the material is available or not. If the material is not available, whatever the research we have done, or it is an endangered species, whatever the research we have done, it will not be that much useful because there is no availability. Very few plants are present. So it is not feasible to isolate large quantities of the natural products. So we have to know whether the plant material is available or not, or whether the microbial species is available or not. And the next, selection of the plant based on the literature or ethnobotanical knowledge or own observation. Yes, when you want to isolate a molecule from a plant, first you have to know whether it is available or abundantly or not. Next, we have to go through whether what type of molecules are present in it. In Ayurvedic text or some other text, whether this plant is described or not. What type of effect of this plant is described. In what way this plant is useful. Whether this information is available or not, we have to surely we have to thoroughly check all the texts and scientific reports. And then next, selection of compounds present in it, and literature survey whether there are any alkaloids. After selecting the plant, after knowing the availability of material, now select the compound. Whether you want to isolate alkaloids or terpenoids or some fatty acids or like that, you have to select a compound. Then only we have to go. We cannot simply isolate whatever the molecule that is available in the plant system. We have to know through texts or scientific reports, we have to know whether this molecule is already isolated or already synthesized, or whether it is having any complex structure, or whether it is a simple structure, how many compounds are present in the plant system. Like this, all information you will come to know through the literature survey. Whether there are, if you are interested in alkaloids, the method of isolation will be different. If you want to isolate, if we want to isolate that means low polar molecules or fatty acids, the method of isolation will be different. So for each type of molecule, there is a different type of method of isolations. A single method is not applied for every molecule. So for that, we have to study the literature survey. Next, study of the structure of the molecule. Yes, after selecting a molecule, that means if we have selected a alkaloid, we have must and go we must and should go through the knowing the structure of the molecule. Yes, if there is any nitrogen present in it, is it an alkaloid? Is it a benzene ring structure is present? Is it a fatty acid or a saturated molecule? Like that, this information. Why we have to study this information is, if the molecule is simple, if it is a saturated fatty acid like that, then it is quite a bit easy to isolate this molecule. If it is a very complex molecule, then the method of isolation will be changed. For sure, this is a very strong basic point. Basing on the structure, you select the molecule. If the molecule is having a very big structure and very complex structure, you change the method of isolation of these molecules. So, first is select the plant, availability of the plant, next select the compound, next go through the structure of the molecule. If we study the structure of the molecule, this analysis and this knowledge will be useful in enhancing the molecules. The type of esters or precursors are used in the media, and we can enhance the amount of the compound. For unknown molecules, or if you want to isolate an unknown molecule, then what we have to do? We must and should do some research by using the techniques. What are the techniques that are useful to isolate an unknown molecule from an unknown plant? The techniques are also given in this lecture. And knowing the structure, in what way knowing the structure of the molecule is, what I am stressing is, knowing is more important. Why we have to study the structure of the molecule? Many natural products have very complex structures. Yes, it is true. These are not synthesized outside. Many compounds, about not even 90% of the natural molecules are very difficult to synthesize in outside environment other than the plant system. So what we have to do is that we have to design the experiment. If the molecule is abundantly available in the plant system, no problem. We can go for it and we can proceed and isolate this molecule. The perceived complexity of natural product is a qualitative matter consisting of consideration of its molecular mass, particular arrangements of the substructures, functional groups, rings, etc. Yes, when you are studying the structure, you identify whether there is a functional group or a ring or something, whether nitrogen is present or sulfur is present or whether any element is present. So studying these structures is very, very useful. Next, the stability of these groups and molecules as a whole, the number and type of stereochemical elements. Yes, some of the molecule it's very difficult to isolate basing on the stereochemistry and the physical properties. If the molecule is a very sensitive molecule, definitely for high temperatures, these will be degraded.
Sorry.
All these viewed in the context of novelty of structure and whether preceding related synthetic efforts have been successful. Yes, when you select a molecule, when you select the structure of the molecule also, we have to know that whether the synthesis of a particular molecule is possible or not. Sometimes it is not possible to synthesize. That type of molecules you can leave and you can again go for another molecule. For that, to proceed the isolation of the molecule, that type of work, we have to study all these things. knowing the availability, knowing the structure, knowing the plant, and also the functional groups present in it. This study will help us whether to proceed the isolation of the molecules in this in particular plant or not. And their very basic point is not all natural products are amenable to total synthesis because it is very cost effective. Many molecules are like that. So it is better to isolate the molecules abundantly available in a plant system. And next, for your better understanding of the structural analysis of the molecules, I have given four examples, a well-known examples, and here you can understand why I am stressing the study of structural analysis. Here we can see colchicine, 3D-methyl colchicine, gloriosin, and taxol. These are the four molecules, and three of the molecules are present in Gloriosa superba, and one of the molecule in Taxus baccata. We can clearly observe here a methyl group is present downside here near NH. Here the methyl group is present in colchicine, and in 3D methyl colchicine, a OH is present here. OH moiety is present, and gloriosin, instead of methyl group here, I am pointing out aldehyde group is present. So these three molecules are available in Gloriosa superba. And one point you have to know out of this slide is already colchicine is available as anti-gout, anti-arthritic molecule, and now scientists are synthesizing this molecule. But coming to 3D-methyl colchicine and gloriosin, these two molecules are not yet synthesized. The structure of these molecules is same except this aldehyde group at NH and this methyl group and this methyl group and this OH. These two molecules are not yet synthesized because of not yet synthesized, and no trials has been taken place because these two molecules are very difficult to identify in the plant system itself. It's quite a it is colchicine is we can easily isolate this molecule, but in order to isolate 3D methyl colchicine, 3D methyl gloriosin, and gloriosin, these molecules are very difficult to isolate and identify this molecule in the plant system. Why? Because the structure is completely all the structure is similar, but except at some points this structure is varied. That means a different moieties are present at one point or other point. Because of these structural differences, it is quite a bit difficult to isolate or identify these two molecules. And for the first time, these two molecules, 3D methyl colchicine is already available. 3D methyl gloriosin and gloriosin are the first time available in India, and these two molecules are already proved to be anti-cancer. And due to unavailability, entire work on these two important molecules has been stopped. Since 70 years, many scientists in India are trying to isolate gloriosin, but not yet. But IBCT will definitely help you and provide you this molecule to trigger further research on gloriosin. And here I want students to concentrate on this structure, a very important molecule, that is taxol from Taxus baccata. You have seen the structures. These are not that much complicated like taxol. Here see the complex structure of the taxol. When you have seen this structure, you can plan the method of isolation in a different way. The isolation of the colchicine will be different from the isolation of the taxol because of the complexity in the structure. So before taking up the isolation of the compounds, just study the structure of the molecules and analyze what type of functional groups are present in the molecules. Basing on that, you select the molecule and go further. Simply if you select taxol and do means, definitely it will not be so easy to isolate a taxol from the plant systems. You have to design another method to isolate this molecule. So knowing the structure of the molecule is very important to plan the experiment on the isolation of the molecules. For your understanding, I have given a well-known examples. And steps, other step. First, how we go? Yes, we have selected a molecule. What we have to do? First, collection of the material. For isolation of the molecule, the next step is collection of the material and powdering the shade dried material. Yes, I will show the live examples here. This is the powder of Gloriosa superba, and we have to shade dry it, and we have to shade after shade dry it and powder it like this. Any natural product we have to get this position. That means shade drying. If you dry in hot sunlight, definitely some of the compounds will be degraded. And and next, immersing in suitable solvents like hexane, methanol, or chloroform. Yes, I want to show another example. That is, see here, this is the Gloriosa superba root powder is immersed in methanol solvent. Like this, we have to immerse the solvent, immerse the plant material, dried plant material in the solvent. And next, a very important technique we have to know before isolation, all this we have to know. Thin layer chromatography to standardize the solvent system for isolation. Generally, if we want to isolate the plant compound or a single molecule in more quantities, definitely we have to use column chromatography. In order to isolate the molecule in column chromatography, which is solvents will be very costly, we have to plan the experiment. We have to standardize the solvent system. I'm not talking about solvent system, because it is not the matter of single solvent. Any compound, the solvent system varies. Using chloroform and methanol is one solvent system, or chloroform, methanol, and hexane another solvent system, basing on the type of the material. If you want to isolate a fatty acid, you can use just hexane. You can isolate the molecule with hexane or hexane with ethyl acetate. If we want to isolate alkaloids, definitely we have to use methanol. We have to develop the solvent system. How we develop, how we will come to know this solvent system is useful to isolate this molecule. That methodology is standardized on thin layer chromatography. Yes, I want to show. This is thin layer chromatography, means we will spot the compound, we will spot the crude extract on the plate like this. This is a thin layer chromatographic plate. Silica is coated on the aluminum foil. We have to spot this underneath here, the crude extract, and we have to develop the TLC plate in the solvent system what we design. So for this, we have to do trial and error method. That means we have to use, in order to develop the TLC, we have to use hexane from low polar solvents to high polar solvents. Not if we use just a high polar solvent, definitely all compounds will move up. So we have to go from low polar solvents to high polar solvents. That means in between we can use different solvent systems. Hexane, ethyl acetate, chloroform, or ethyl acetate chloroform, like that, basing on the type of the material and type of the compounds present in it. And this thin layer chromatography is not only in one way it is useful. Thin layer chromatography is useful to standardize the solvent system, to know whether how much amount of the compound is present in the system. If the band is thick, we can analyze that this molecule is more in the plant extract. And to know that, and to know the retention factor of the molecules, and to know the the information for your better understanding in next slide. Next, for isolation of the compounds, we use generally column chromatography. I will also explain that. And after after column chromatography, next is, before that, there is another technique called preparative thin layer chromatography. That means if we want to isolate a molecule in small quantities, definitely that means 10 mg, 20 mg, we can do this experiment on preparative thin layer chromatography like this. And you can see the bands here, very clear bands are here. Every molecule is separated. If we want in less, madam, am I audible, madam?
Yes, yes, you are
Yes,
audible.
madam.
Ah,
Yes.
yes, madam. Yes, madam. Audible, madam.
Proceed.
Yes, madam. Yes, madam. TLC visible, madam? TLC visible?
Not actually. It is not very clear.
No, madam. No,
Yes,
madam.
madam. Yes, because there are many compounds in it. Many
Okay.
compounds. That's why the TLC is like in Gloriosa Superbra, at least minimum on this TLC plate around 30 to 40 compounds are there.
Okay.
That's
Okay.
why very congested.
It's okay. Proceed, madam.
Uh, yes, madam. Madam, this one is a visible plant material, everything.
Yes. 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 It is visible.
Okay. I will continue, madam. Yes, next is column chromatography, and in column chromatography, if we want more compound, that means 3 grams or 4 grams. If we need to isolate, means we can use column chromatography. If we want less quantity, then we can use preparative thin layer chromatography. And there is another. This is another TLC, and here the concentration of the compounds is less. That's why TLC is somewhat white in color. Here bands are clear. And thin layer chromatography is what are the uses of different techniques involved in the isolation of the compounds. Not only after isolation, also there are different techniques how we analyze the compound. First, we use thin layer chromatography to standardize the solvent system. Next, preparative thin layer chromatography to isolate single molecule in small quantities. And next, after isolating the molecule through column chromatography, next we have to submit the molecule for spectroscopy. That means to know how many protons are present, how many carbons are present, how many oxygens are present, or how many functional groups are present. To know all this information, we have to submit the molecule for spectroscopy, that is proton NMR, 13C NMR, molecular weight, GC mass or LCMS mass, and all these spectros. To know the structure of the molecule, we have to submit the sample to the proton NMR initially, first proton NMR, next molecular weight spectroscopy. And, and another technique that is involved in this natural product technology and isolation of compounds is high performance liquid chromatography to know the purity. Yes, we use this HPLC to know how many compounds are present in the extract, and also we will come to know how much amount of the compound is present. If it is 1 mg or 2 mg, we will come to know basing on the data, basing on the statistical analysis, basing on the retention time and peak area and everything in the HPLC, we will come to know how much amount of the compound is present in the plant extract we have submitted for the HPLC spectrum. Yes, next after this is gas chromatography with mass, GCMS, to know the number of compounds in the extract. Yes, when we want to isolate a molecule, initially we will come to know. Initially, this is another technique that is used to identify how many compounds in my plant is there or there. Definitely, if it is, we will come to know with molecular weight combined, coupled with the GC gas chromatography, will come to know the structure of the molecules also. If the molecular structure is unknown, definitely we will give the spectrum itself gives the this molecule is unknown with with that molecular weight. So this technique is useful to know how many compounds are present in the extract. And another technique that is involved is LCMS, that is liquid chromatography coupled with mass. That means by using the liquid, the compounds are on the stationary phase. This liquid phase will definitely move the compounds up. With this LCMS, we will come to know how many compounds are present in the extract. But these two techniques are similar, gas chromatography and also liquid chromatography. But the only thing is here, gas is used as a mobile phase, and here liquid is used as the mobile phase. And another simple technique, that is paper chromatography to separate the molecules. This is along with thin layer chromatography, the same principle is used here. This is another simple technique. So all these techniques are involved in isolation of the molecules. If you want to isolate, definitely we have to go through basics of these spectroscopies and chromatographies and how we isolate. Then only we will be successful in isolating or discovering a drug from the plant system. Without this information, if we blindly go, means definitely money will be wasted, and definitely there will be Herculean task, our hard work will be wasted. And next process, plant material is collected and identified by a taxonomist. How we will do that? If we have selected a plant, what is the procedure for that? And we have to identify, we have to go to a taxonomist, and we should ask him whether this plant belongs to some brassicaceae or annonaceae or euphorbiaceae, like that, because many plants will look alike. We will get confused by seeing the nature of the plant material and height, length, everything. So we have to take the help of the taxonomist before selecting a plant. Next, then it is shade dried and powder. Yes. Next, powder is immersed in suitable organic solvents. This is the most and important point. Why I am stressing is, if we want to isolate alkaloid, definitely we have to put the plant material in the methanol. If we put the plant material, if we immerse the plant material in hexane, definitely it is not possible to identify the alkaloids in the low polar solvent like hexane. For that reason, we have to compulsory know which type of organic solvent is used to extract the molecule from the dried, shade dried plant material. So generally used is DCM methanol and methanol in 1:1 ratio. This is an appropriate solvent system to isolate the molecules, to extract the molecules from the plants. Then after a few days of soaking, the solvent is filtered and concentrated in rotaevaporator, which is now called crude extract. Yes, after filtration, now this is called, we have to concentrate in rotaevaporator, then it is called a crude extract. Crude extract is powdered by adding silica and loaded in silica column and eluted with suitable solvent systems like hexane, ethyl acetate combinations. I have already stressed this point that hexane and ethyl acetate is a very good solvent system. With this solvent system, we can isolate different compounds, different combinations. And compounds in the crude extract mixture is separated one by one, which is due to retention factor. Yes, when we are going to isolate the molecule on a stationary phase. Stationary phase is nothing but the silica powder. And with and next thing is the mobile phase is solvent system. What we apply the solvent system, that is nothing but with this help of solvent system we will isolate the molecule. How the compounds are isolated is based on the retention factor. How much time it is spending in the column with the stationary phase. How much time it is how much time it will take for a molecule to come into the mobile phase. Basing on this retention factor, compounds are separated in the column chromatography or in the thin layer chromatography. The eluted compound is tested on TLC. Whether the compound is showing single spot or multiple spots, we are going to know this one. Yes, you can see here. Yes, this is the thin layer TLC plate. We can see here single spot, that is colchicine, and here the crude extract, and here in this line is the crude extract. Many compounds are present in the crude extract, but I have spotted here single molecule, that is colchicine, like that. So this thin layer chromatography is useful to separate the compounds and to know the retention factor of the compounds with the help of mobile phase, that is a solvent system. Then it is called, if we isolate, if you identify the molecule is a single spot, definitely then only we have to give for HNMR. Even if we give crude extract for HNMR, it is difficult to identify which proton belongs to which compound. So pure molecules will be separated on the column or on TLC, and we have to submit this to HNMR or 13C NMR. Initially, we go for generally HNMR, proton NMR. Then we will go for, if the molecule is pure, then we can submit for 13 NMR. We cannot encourage to submit students to submit the crude extracts for HNMR or 13C NMR. It is quite highly difficult to elucidate the structure with crude extracts. A pure compound is also tested on different then after submit, then it is called pure compounds. Then these pure compounds are tested on different cancer cell lines or other microbes or other diseases. It is better, instead of a crude extracts, it is better, it is encouraged to do the experiment on with the pure compound on different diseases like cancer or microbes or infectious diseases, whatever we are interested in. And in this, for better understanding, I have given in pictures the information, that is plant material. This is Gloriosa superba plant material. This the tubers of this plant material is here, and how the solvent is poured is dry material. How the TLC takes place here. the small experiment, these compounds, how the compounds are separated on TLC plate. Just for understanding, I have given this information. Here the roots of Gloriosa superba, after drying they will look like this, and after powdering and putting solvent it will look like this. And next, thin layer chromatography. As I have also already stressed at this point, very important basic technique. Some of the solvents I am using, here we have to spot like this, and then after spotting on the thin layer TLC plate, we have to develop the plate in TLC chamber, and then after that we have to observe under short UV light and also long UV light, because this is a way observing under UV light is gives us very good information whether the molecule is long UV active or short UV active. Generally fatty acids and everything low polar molecules will not be visible on TLC plate. So very good alkaloids or some terpenoids or pharmaceutically bioactive molecules, definitely they are visible under short UV or long UV light. This is an important step. After developing the plate, we must and should observe under short UV light. The TLC plate I have observed and taken photo under short UV light. It will look like this. The left side lane is crude extract of Gloriosa superba, and the right side lane, this big spot is colchicine. It will look like this. The plate will look like this under short UV light. Then we will come to know whether this, even in colchicine also some other compound is present. We will come to know with on TLC, only observing under UV light. And when we see, when we generally the TLC plate in visible light will be like this. When we put under short UV light, definitely you will see the spots. So for better understanding, I have given this photo which is observed under short UV light, and I have marked. If you want to isolate the colchicine molecule, I have marked it like this with pencil. We have to mark with a pencil that this is colchicine, and at the same retention factor, at the same here horizontally, if you see in the crude extract also this molecule is present at the left side lane. This molecule is present. This also indicating where the molecule stops. Also at that point only here in pure molecule extract also definitely this molecule stops there. So we can identify these two molecules are same, and we have to mark it, and we have to, then we can design the solvent system. At what solvent system this molecule moves to the middle of the plate. For this, for moving the colchicine to the middle of the plate, I used chloroform 95% and 5% methanol. In this solvent system, this colchicine moved to the middle of the plate. This solvent system is appropriate, and we can isolate with this solvent system this molecule. So to know that solvent system, we have to put TLC. And after another step, after identification in the UV light, we have to put it in the iodine chamber here for absorption. So here this is the silica powder with iodine, and the TLC plate I have put in the bottle consisting of iodine in the silica. See, the brown color powder is iodine with silica. We have to definitely, this is an important step. Why we have to do this step is to know, to visualize the spots. Some compounds after adsorption will be, we can spot. The colchicine as a brown color spot here. And after that, in the methanol reagents, definitely we will visualize on the compounds. If we use dragon orange color or yellow color spots, here I have used, in this TLC, I have used methanol sulfuric acid. So the colchicine here turned into yellow. After dipping in methanol sulfuric acid, the colchicine turned into yellow, and here that spot is also present here in the crude extract. So there are different reagents. We cannot depend on single reagent to visualize some other compounds with phenolic compounds. We can use potassium permanganate solutions. In that, the phenolic compounds will be visualized as orange color or bright yellow color compounds. Here you can see after spraying methanol sulfuric acid, this is looking as yellow color. And next, after that, I have to char on the hot plate. We have to put this hot plate, and this is the pure color. Charring like on the for all colors and everything. If we dip it in a drag and drop reagent, definitely this will turn into orange. For terpenoids or fatty acids, definitely they will turn into brown. So basing on the colors to visualize the spots, we have to spray the reagents correctly. And yeah, I have given the picture here. This is the preparative thin layer chromatography, and this is the column chromatography. You can see here how the compounds are separated. Here, this one blue color compound is here, and here first orange color. This is coming down with the one type of solvent system. If we use completely methanol, all compounds will come down. And this is another photo. Here all the yellow color compounds, and here extract black color one is nothing but the plant extract, crude extract. And here one black spot is here. This is also another compound. Through column chromatography, we can separate the molecules like this. But always compounds will not be visualized in the column. Some compounds will be invisible. They are white. So they will not give any color. We cannot judge that the compound is not there. Even in the right side column also, there is a compound here also. But it will not be visualized in the column. That spot will be visualized in iodine absorption or after spraying the reagents. So most of the compounds are like that. Only very few compounds are visualized in the column. And after that, for a better understanding, I have given the spectros also. How before isolation of the molecules, we can give to the gas chromatography GCMS. This is the GCMS how it looks. And here in extracts there are many compounds, and we can visualize here the molecular weight is not given, but that type of spectra is also there, and we can submit GCMS also. And here LCMS mass, this is an NMR, not LCMS, this is an NMR, nuclear magnetic resonance spectra, HNMR. This is not LCMS. And this is HPLC. All these spectra belongs to my research work only. It is not copied from the internet. And this is the HPLC spectra, and how it looks, the peaks, how it looks. Here the area of the peak it is occupying, and the retention time is given here. Basing on this data, we can analyze how much amount of the compound is present in the plant extract. Basing on this GCMS spectra, we will come to know how many compounds are present in extract. And here in an HNMR, this we will come to know how many protons are present in the compound. And this is an IR spectra. With this IR spectra, we will come to know what are the functional groups, OH or NH or CHO aldehyde, or whatever the functional groups that are present in the molecule, we will come to know with this IR spectra. And these are some of the examples to get a very good imagination or observation. I have given this. You can go through these spectra once again. And another, after testing, after analyzing the molecules, yes, you got a very good molecule that is an alkaloid. You have isolated the molecule. Then you can test it on microbes. This is the antimicrobial activity of one compound isolated from a duckweed, that is an aquatic plant. This work I have done, already isolated an antibacterial molecule. And here, see, you can see there is no growth of bacteria around this disc, white disc. There is no growth of bacteria, clearly indicating that the molecule is antibacterial. Here with the disc method, antimicrobial activity we can test. This is this is how we have to test the antimicrobial activity with the disc method. For your understanding, and you will get a very good picture. That's why I have given this photo. And next, not only testing on microbes, but also you can test the pure molecules on cancer cell lines or other diseases. For example, one thing I have given, and another one is here testing on anti-cancer activity testing. This is one protocol how we test the secondary metabolites or bioactive molecules on the cancer cell lines. And here this is one type of activity, and this is another type of activity. A compound may have many properties. It may defend nutritional, it may defend microbe or fungi, bacteria, or some other marine organisms or snails or fishes or whatever it may be. This type of one activity we can do, another type of activity, and there are other diseases we can test. And what are the challenging features? Yes, there are also some bottlenecks in the isolation of the natural products. What are the challenging factors in this? The main challenging factor is the availability of the molecule in the plant system itself is very, very less. It will not meet the public demand. This is the very important point. And many species are endangered species. Example, Taxus baccata. Taxus baccata is a very good anti-cancer molecule producing plant, but this plant is an endangered species and the availability of the plant is very less. So what this is the current scenario of the many medicinal plants, not only one. And this is another challenging factor to isolate to produce a antibacterial or antibiotic or bioactive molecules. And next, the compounds isolated from plant or animal sources cannot be isolated in large quantities. Yes, it is true. Even if we take 100 kgs of materials also, many bioactive molecules will get in milligrams. So that this is a challenging factor. So we cannot, for to meet the public demand, we need cages and cages, but we will get Taxol is also like that. So what they will do is they will isolate the molecule till one step, and then they will they remove that molecule at an younger age of the plant, and they will semi-synthesize. They will add some functional group like that, and they will produce the pharmaceutically useful compounds. So in large quantities, we cannot isolate any natural products. For that, to isolate the molecule in large quantities, there are other technologies. To overcome these are the drawbacks, and to overcome these, there is genetic engineering technology. Yes, now comes the genetic engineering technology to enhance these molecules in the plant system itself or in the bacterial system. The natural product structures are complex, therefore, many isolation and separation techniques. This is one challenging factor. Because of the complex nature of the natural products, quite a bit difficult to isolate these molecules. Yes, these molecules comes from the column combined with another molecule, another fatty acid like that. That is the nature of some of the natural products. An example is vincristin and vinblastine. Vincristin and vinblastine is also having a very complex structure. So we have to design the method definitely before isolating these molecules. And next, accessing sufficient biological material to isolate and characterize bioactive NP may also be challenging. Yes, availability. Not all plants are available in our surroundings. Many plants are in some hill regions or some beside seashores like that. So it is not accessible to collect the plant. So these are some of the challenging factors to isolate the molecule that we encounter for the isolation of the molecules. So in order to overcome these challenges, what we have to do? Now comes the picture, now comes the subject, biochemical engineering or genetic engineering or biotechnology. These three subjects will come forward now to enhance these molecules. For the drug discovery, column chromatography, thin layer chromatography is useful. But to enhance the molecule to meet the public demand, now comes the genetic engineering and biotechnology or biochemical engineering. And, and how to overcome. Large quantities of bioactive natural products are produced in fermentation cultures by microbes. Yes, through fermentation, it is also well-known technique. Every student knows that through fermentation, many of the molecules are produced by the microbes. And there is, during fermentation, some chemical reaction takes place because of the enzymes released by the microbes. That is one type of technology producing bioactive natural compounds. And another technique is growing callus cultures or hairy root cultures. Hairy root cultures comes under genetic engineering technology, that is transferring the genes from bacteria to plant system and making the plant to produce more amount of compound. This is what exactly is hairy root induction. We will infect the plant with bacteria. Then now the plant system, the metabolism of the plant will change after the infection of the plant, and definitely because of this infection, now the plant is able to produce more amount of compounds. Example I want to give is Withania somnifera. Withania somnifera produces very less amounts of bethaferin A or withanolide A. But what happens is, when it is produced in low quantities, what we do is we will infect the plant with microbe. We will infect the plant with agrobacterium rhizogenes. Then the plant is able to produce more hairy roots. More hairy roots also means, that means unlimited growth of hairy roots. Hairy roots are produced from the infected region. Unlimited growth means unlimited amount of the compound. That means we can obtain more amount of the compound by using more amount of the hairy roots. This is an unlimited cancer growth. It will not die. Generally, plant dies at the age of many plants like 6 to 7 months. But now hairy roots, that age limit is not there. This is a unlimited cancer growth. I will show in pictures also how the hairy roots looks. This comes genetic engineering technology and genetically modified plants. Yes, we will modify the genes in the plants to produce biosynthetic genes. That means there for synthesizing many molecules there will be a pathway. In the pathway we will modify some of the genes. We will modify it. Now the plant is able to produce more compound. And next, tissue cultures. Next, callus cultures. Callus cultures is nothing but another modification. With agrobacterium tumefaciens, we will infect the plant. Now the plant able to produces cancer-like growth. Now we can isolate the molecule from these callus cultures. Next technique is how to enhance the molecule is culturing with help of media manipulations. Yes, after genetic transformation also, many plants use less amounts. What we do, we will, when we study the structure of the molecule, if it is having a benzene ring, that type of enhancers or that type of elisters are used in the media, and definitely the compound will be enhanced. That means 1 gram. Generally in plant system, in vivo system, the plant gives 1 gram. By using this type of media manipulations, we can enhance the molecule to 10 grams like that. Now we can meet the public demand by culturing these hairy roots or callus and everything in the bioreactors to get large quantities of the compounds. Generally, in natural system, very less, but by using genetic engineering technologies, we can enhance the molecules. And these are some of the technologies, not even one technology. Here, fermentation culture and growing the hairy roots, or genetically modifying the plant by modifying the genes and everything, and tissue cultures, that means in lab cultures, culturing that particular tissue, and callus cultures, that means creating a cancer in the plant, and media manipulations. This media manipulation helped me to enhance vitaferin A to 24 folds. During vitaferin A, I have increased it to 24 folds because of the media manipulation. In one type, when I have supplemented the media with one type of carbohydrate, it enhanced it to 24 folds. That means that particular enhancer or elicitor or whatever the molecule I have added in the media enhanced this particular vitaferin A. So that I have identified through HPLC, and that HPLC is already here. These two peaks are, one is vitaferin A peak, one is vithanolide A peak. This here we can see how we can standardize. I have standardized the method to identify this molecule in the HPLC system. With the help of this, we can give the crude extract to the HPLC, and we will identify how much amount of the compound is enhanced, whether it is enhanced to 24 folds or 30 folds or 1 fold. So with the help of HPLC spectra, we will identify whether the compound is enhanced or not. This technique is used to identify the enhancement. So that's why all these spectra are given for your understanding, and the technology. And I will come. Yes, yes, culture media manipulations, callus cultures. And these manipulations are very useful throughout the world. This type of technology is used now also to get some compounds like vitaferin A or vithanolide A or taxol or something else. These media manipulations are scientists are doing these media manipulations and enhancing the molecules. This is another important step. I have given one example on ashwagandha. And steps involved in enhancement, and what is in genetic engineering technology. This comes under this whole part is nothing but a single technology I have given. This is genetic engineering technology. How we do? Actually, we will culture the plant in the lab like this. Then what we will do is we will cut the leaf tissue and put in another bottle. Infect the plant. I have infected this leaf tissue with agrobacterium. Now the leaf is able to produce hairy roots. Now the metabolism changed. The place where the infection has taken place, at that point, here hairy roots are growing. This is an unlimited growth. For a plant, it is a limited growth, but here hairy roots are unlimited growth. So you can see here clearly arising hairy roots arising here in the leaf tissue. This is called an explant. This leaf tissue is collected from here, which I have cultured in the test tubes or bottles in the lab. It is not an in vivo plant. This is an in vitro plant. And next, these hairy roots are cut and again subcultured in a media to get more cultures. Here, see, this same thing is cultured in very, very big reactors, and more hairy roots are produced and more amount of compound is isolated. This is a genetic engineering tool, and also we can, for the especially pharmaceutically useful natural products, this technology is used in many foreign countries, and IBCT is now trying to do this type of experiments on many medicinal plants. And IBCT will help you in guiding how to do this genetic engineering, how to induce the hairy roots in the medicinal plants. We can use this technology in enhancing other nutritional compounds. That means if you want to, if there is any nutritional factor in some medicinal plant, we can use this technology. But for pharmaceutical needs only, many of the scientists are using this technology. And here you can see the callus culture. This is cancer-like growth, and with the help of Agrobacterium tumefaciens, we can create cancer in the plant, so that this unlimited growth will give more amount of the compound. Sometimes more material, more compound, or sometimes metabolism will be changed and we will get more amount of the compound. All this for your for a better understanding I have given in the pictures. And hairy root cultures in different media, and I have already said that media manipulations are very important. After genetic transformation also, sometimes you will get less amount of compound. I have used one type of carbohydrate here. I have used some sucrose, and in this bottle I have used glucose, and in this one fructose, and this one maltose. But when coming to the glucose here, the growth of the hairy roots are more. That means using glucose in the media definitely gives more growth of the hairy roots. That means this is enhancing the growth of the hairy roots. But enhancing the molecule in the system, we have to identify through the HPLC. This is the media manipulations for this. You can see this picture and you can identify and you can understand that how the media manipulations will help to enhance the molecule in the plant tissue or growth of the tissue. And here, after that, after enhancing, after culturing the hairy roots like this, there is another technique called DNA isolations and plasmid isolations. We have to identify whether the gene from bacteria integrated into this tissue or not. To identify that, DNA, RNA isolations and plasmid isolations we have to do. This is one technique. This is another big genetic engineering technique that we can use in enhancement of molecules. Not only organic chemistry, here you have already learning that genetic engineering, also biotechnology. Here this isolation comes under biotechnology. And here a very clear picture of this hairy roots growing in the media and supplemented with this one gelrite. Not always agar, we can use gelrite also to visualize the hairy roots here clearly. And after that, whether the DNA of the microbe is transferred in the tissue or not, we can identify it with the help of PCR, polymerase chain reaction. Here this is picture is also my research work, and every picture is my research work and not copied. And here this is the DNA isolations, and whether the gene is present in the tissue or not. Here I have given these two fluorescent bands are nothing but the enhanced gene through identified through PCR, and this is nothing but the gel where the DNA is present enhanced. With the help of PCR I have identified this. This is another technique in the biotechnology. That means altogether organic chemistry, physical chemistry, genetic engineering, and also biotechnology is involved completely in the natural product technology. And sometimes and another technique that is very important, well-known technique is fermentation that I have given. And this is, everybody knows this topic, fermentation, a chemical reaction takes place. Another important point, a very basic and new point I want to give information is that not all plants are able to synthesize molecules. There are some fungal species or bacterial species involved in the synthesis of the bioactive molecules, not in all plants, but in some plants. For example, recent reports are clearly saying that taxol is produced with the help of endophytic fungi. With clear live example, I want to give. See here. This is Gloriosa superba root powder. In this root powder, there is fungus growing in it. In this fungus, this fungus is also helping the plant system to synthesize the particular colchicine or gloriosin. So definitely there are mechanisms that microbes are helping the plant in the form of symbiotic relationship to produce natural products. This is a very new point I want to say, and you can see even in Gloriosa superba also fungus is grown. That means this fungus may be endophytic fungus. So they maybe this fungus is helping the plant to grow to synthesize the particular colchicine or 3-methyl colchicine, whatever the active molecule may be. So I want to students to remember this very basic point and new identifications. Very every scientific reports mentions this point, but we just use fermentation only. But this is another technique. So how come this technique is useful in enhancing the molecules? I will tell. When the plant material is not available, this fungus is taken and cultured. This fungus is taken out and cultured in the culture system in a broth, and definitely this fungus will also produce colchicine, gloriosin, or 3-methyl colchicine. In this way, we exploit the symbiotic relationship of the plant and microbes to get the more molecules. That means if the plant is producing 1 gram, if we isolate, if we culture bacteria or fungus, definitely we can get more growth and more compound we can get. In this way, definitely the challenging factors what I have said, we will overcome that factors, that now we will get more compound to meet the public demand. So this is a very good example I have cultured since last week. You can see the fungus. and and this is a very important technique. Another method, this is a feasible technique to isolate the compound of interest. Why I am saying feasible? Cultivating the plants in acres and acres, now it is not possible if there is a land deficiency to cultivate the medicinal plant. Now we can in labs itself, we can liters and liters of microbial broth is microbe is cultured in broth, and definitely by it will release the compound into the media, and definitely we can get the natural product out of this media. So now it is not now acres and acres of land is not required. Now fertilizers are not required. So that's why that's why this technique, this endophytic relationship of plant and microbe is very useful, and we can exploit this technique for isolating good natural products. It is true that recent identifications are that endophytic fungus in the plant system is helping to produce taxol. Not only one plant, many plants like that. Even in Gloriosa, there are reports that fungus is helping. And isolating the bioactive molecule which is synthesized by the microbe. Microbe will release that compound into the media, or by by or by killing the killing the bacteria in the broth. Definitely we can isolate that molecule. So these are some of the techniques apart from the fermentation. Fermentation everybody knows well. And here you can see in picture I have given here, and see here in the live picture I have already shown. This is the fermentation, and this is the culturing the endophytic fungus. We will isolate this fungus from the plant system itself. No need of getting from the soil. But if we cut the plant material and if we grow in the media, definitely some fungus will grow. That we can say that this is an endophytic fungus. So we can exploit this technology for the use of. This also comes under biochemistry and biotechnology. And after learning all this, I can suggest, IPCT will suggest, because there is an immense potential, and even every student after completing MSc chemistry or MSc biotechnology, by learning this technology, you yourself can establish small self-employment, micro scale or small scale industries. Definitely there is an immense wide potential. Now you can isolate any molecule from any plant, definitely for sure. And it is a self-employment opportunity for any person to isolate a natural product and supply to the markets or pharmaceutical companies. Now pharmaceutical companies are showing some interest in natural products. Now you can do that after going through all this isolation of molecules techniques and biotechnology. You can plan the experiment, whatever the products that are useful for the society. You can definitely isolate these molecules and supply to the pharmaceutical companies. No need of again and some techniques are with less capital. I will tell you how, what are the uses of learning these technologies. At current pandemic situation, now the research is enhanced in the isolation of compounds or natural product research. It has been enhanced throughout the world, not in India. And there is a tremendous scope to establish micro scale or small scale or large scale units to produce natural ingredients. Yes, many companies are asking that we want that product or this product. Yes, one of the example is vithanolide A. Vithanolide A is anti-cancer molecule. This anti-cancer molecule which cures the neurological problems. Yes, many companies are in want of this vithanolide A. Now you can isolate with the help of these technologies. You can isolate vithanolide A and supply to the pharmaceutical companies. In this way, this technology, learning this technology is useful. Next, few natural products manufacturing units that can be started with less capital. Yes, there are. If you find this is very difficult, it is very costly. Means definitely there are some techniques that are with less capital only. You can establish a micro scale industry and producing extracts or Ayurvedic proprietary medicine. This will cost very less. If you want to supply some powders, that means Vincristin powder or Vinca rosea extract, you can supply, because you can supply Vinca rosea extract to pharmaceutical companies if we don't have that much capital. So another is herbal Ayurvedic proprietary medicines. That means in Ayurvedic text there is a protocol how to extract the molecule. We can take that procedure, and we can, in the same way, you can prepare some extract of some medicinal plant described in Ayurvedic text, and definitely with the Ayush department will give permissions with less certificates, and you can also prepare Ayurvedic proprietary medicines. And next is herbal soaps and detergents. Yes, this is recently herbal soaps gained lot of importance, and a very big market is there for herbal soaps and detergents. You can prepare, isolate this extract, not single molecule. You can directly extract, isolate the non-harmful plants extracts, and you can use this extract in herbal soaps, and you can export also. Permissions, we will get the permissions from the Ayush departments or in the present in our own states. And production of biopesticides, this is an important plant because many cancer diseases are arising due to pesticide food we have taken. So in order to avoid that, in order to avoid this problem, we can use biopesticides. That means extracts of plants or extracts of microbes or extracts of mangroves or marine organisms, like that we can extract, and we can test it on different microbes that are affecting the plant. So you can test this on poultry also. You can extract the molecules from the plants, and you can test it as a medicine for fishes, and poultry also you can test. In that also a lot of scope is there. Even if you establish a micro scale also with less capital also, these biopesticides and poultry is flourishing now. You can market these compounds. You can isolate, you can extract, and market these compounds also with less capital. Small scale setups include pharmaceutically important compounds. Yes, if you are more interested in very good compounds, like if you want to supply colchicine also, with these protocols and everything, you can isolate this molecule and in less amounts. Initially, in less amounts also, we can supply. That type of opportunities are there in India. Lot of opportunities. Just thing is learning the technologies and going forward. Learn all these technologies and go forward. And definitely, IBCT will help the students and research scholars and scientists and professors how to develop a small scale industry with the help of natural products technology. Learning all these will create a self employment opportunities in this field. And here I have given the references also how the natural given, you can go through it. And in my thesis also, every technology is clearly described. And I also thank um, the St. Teresa's College, my college. I'm very thankful to everyone me. And I'm thankful to yes, madam. Good.
జ్యోతిర్మయ్య గారు మ్యూట్ లో ఉన్నట్టున్నారు, చూడండి ఒకసారి.
Thank you, Madhavi. We
Yes, madam.
sincerely appreciate you for taking out your time from your busy schedule to provide us with such valuable information and excellent presentation. All the work is practical and easy to have the work, and students are exceptionally benefited from your presentation. I wish you great luck with all your future projects and look forward to work together with you. There are two words that have the power to transform our health and happiness. Gratitude and appreciation are also essential for a healthy work environment. So, I call upon Dr. M. Rama, Associate Professor, Department of Chemistry, to propose vote of thanks.
Good afternoon to one and all. I deem it my privilege to propose vote of thanks for today's national seminar. First of all, I would like to thank God Almighty for His abundant blessings showered upon us. My heartfelt thanks goes to our correspondent and superior, Dr. Sister Marietta Pudota, for her prayers and wishes, and to our beloved principal, Dr. Sister Marietta D. Mello, for her constant motivation, and to our vice principal, Dr. Sister Shauridu, for her encouragement. Thank you, dear sisters. I would like to thank from bottom of my heart to today's resource person, Dr. Madhavi, Institute of Biotechnology, Hyderabad, for her elaborative thoughts on natural products technology, different ways and means to extract them from plants, and their significance in our daily life. Thank you, madam. I would like to thank heads of various departments for their coordination. I would like to thank Dr. C.A. Jyotirmai Garu, head of the Department of Chemistry, for her efforts to make this event successful. I would like to thank the participants from various institutions for their participation, and also the department members of the Home Institute for their coordination and cooperation. I would like to thank dear students for their enthusiastic presence in the seminar, and I would like to thank the technicians and support staff for their timely help. Thank you, one and all.
Thank you, ma'am. Once again,
Yes,
I deem it
ma'am.
a privilege to have Dr. Madhavi with us, one of our alumni. We are proud of you. Our institute is proud of you by establishing a research institute by itself. Hearty congratulations for everything, and for thank you once again for taking your time and presenting an excellent presentation. Looking forward to work with you for research projects, consultancy, and everything. Thank you once again.
Yes,
I also acknowledge the presence of Professor M.N.V. Prasad Garu with us. Sir, thank you for being with us. We also remember all your contributions towards our institute With immense pleasure and grateful heart, I thank you for being with us. I also thank all the students for your active participation. If you have any queries, Madam is with us. You can ask. And the feedback link is placed in the chat box. You can fill the feedback link so that you'll be getting the certificate within a week. Dear students, you are welcome to ask any questions if you have. This is the time for queries. If not, we will conclude the session. You want to say something, Dr. Madhvi?
Yes, madam. Yes, madam. Definitely, I welcome if any question or if any doubts arises from your side, you can contact me through website or on phone call. And I'm very thankful to the institute for providing me this opportunity to give good information to students. This is a platform for me to give you important things. And I also thank Madam Jyotirmayi Madam for even in intermediate also I have learned many things from her, and I'm very thankful to you, Madam. Definitely for giving me this opportunity. Principal sister also, I'm very thankful to her. If
Okay.
any doubts, you can ask me. You can approach me through website or some through phone call also. I can answer, or through WhatsApp also I can answer.
We'll share your website.
Hello.
Yes,
Sure.
madam. Yes. Yes, you can ask any question if you have any doubt.
I heard somebody asking a question. You are free to ask them. Dear students, you are free to ask. So if there are no queries, we'll conclude the session. I
Yes,
thank each and every one of you for your presence.
okay,
Once
madam.
again,
Thank you
thank
very much,
you.
madam. Yes, yes, definitely. You can approach anytime, madam.
Okay.
Okay, madam.
Thank
I
you.
can conclude. Thank you, madam.
Thank you. Tommy sir closed his saying.